Okay, so for this audio lecture, we're going to be covering concepts in Chapter 3, Recognition and Response. So for recognition, we're talking about receptors that will recognize the antigen, and then response, what happens to the immune cell. So we can have proliferation, we can have production of cytokines. Like Chapter 1 and 2, this is a kind of an overview chapter. So we've talked about some of these, receptors, but we're just going to talk about them again just to reinforce and create that foundation for the rest of the semester. So a lot of these concepts that are in this chapter will be expanded upon in other chapters. So just like with Chapter 1, 2, and this chapter, if some of this kind of doesn't make sense, just try to focus on the big picture because the details will be added in through the course of the semester. So first we're going to look at the immune antigen receptor molecules. So the receptors that will bind antigen on an immune cell would be our B cell receptor, BCR. So as we talked about before, the B cell receptors can be found on the B cell. As you can see on the figure towards the left, panel A, that B cell receptor has an antibody structure. And so that antibody, that B cell receptor, would be specific to a specific antigen. So it's part of the adaptive response, the specific response. For the T cell receptor is going to be found on our T cells. And so they're going to be able to bind peptides that are presented by APCs or antigen presenting cells on the MHC molecules. So the MHC molecules would be found on the antigen presenting cells. So a lot of times these are phagocytes, neutrophils, macrophages that have undergone phagocytosis, and then they're going to present that antigen to the T cell via the MHC receptor on their side to the T cell receptor. Again, on the left, on panel there's panel A. We have on the lower one, we have an example of a T cell receptor, and so you can see how it's a dimer. So there's two portions. And like the antibody, there's a variable region. So the top piece that's labeled V varies between different T cells. Okay. We then have co-receptors, these accessory molecules like CD4 and CD8. So these work with the T cell receptor in order for signaling to happen that needs to occur in order for there to be a response. So we want to keep in mind that the principles of receptor ligand binding that occur with any receptor and ligand also occur with our immune cell receptor and its ligand, its antigen. And so we can see in our figure here on the right side, we have our receptor, and it's going to be interacting with the ligand or the antigen that's on the left side. And those interactions are going to occur through multiple non-covalent bonds. And so how many of these bonds occur and what type of bonds lends to how strongly we're going to get binding between the receptor and the ligand, and this is known as binding affinity. So again, binding affinity is how strong the receptor is going to bind to its ligand. Obviously, if we were to change some of the amino acids on the receptor or on the ligand, this could affect binding affinity, either making it stronger or weaker. And one of the things we'll talk about later on is about how these receptors can change. So there's a maturation of these receptors as the B cell, as the T cell matures. And so some of this is by doing point little mutations to the receptor so that it has a stronger affinity. So always the goal would be to have a stronger affinity to the ligand in the immune response. Okay, and so if we look at these individual bonds, we can actually, if we add them all together, we get the cumulative bond strength. And whenever we're talking about binding, one of the Things that are looked at are dissociation constant, or the KD, and that measures the strength of the ligand binding. So another way to change the strength of the binding is by having multivalence. So multivalence is where there'll be multiple binding sites. So in panel B, you can see how the antibody is actually binding to multiple sites at the same time in the middle there. And that would be a stronger binding or have a higher avidity. So avidity is the combination of all individual bindings through the multiple interactions, where affinity is a single interaction. Okay, so when it's univalent, that would be affinity. So ligand receptor binding can induce molecular changes in the receptor. So this, again, these are principles that are, you would have learned in cell bio. So there can be conformational changes that can occur. We can actually get dimerizing or clustering that can happen. This dimerizing and clustering helps to create multivalent ligand binding. So you can see down in the panel below how if we have individual receptors, we're only able to bind, one spot, where if we start to dimerize or even start to cluster, we can get bivalent or multivalent binding to occur, which again would mean that we would have a stronger binding occurring. Other changes that can occur is the location on the membrane. So moving into lipid rafts would be an example. And we could also get covalent modifications. So when these receptors are altered, this can cause a cascading of intracellular events known as cell signaling. So we can get activation of enzymes and changes of intracellular locations of molecules. So we can also get aggregation due to the ligand binding, and this actually will enhance the ligand binding KD. So if we look at our figure, if we only have the alpha strand of the IL-2 receptor, we're going to have a low affinity. If we have beta and gamma, this is going to be an intermediate affinity, but if we have all three together, this actually creates a high affinity to the IL-2 by the receptor. So again, this aggregation of components together increases that binding affinity, and binding affinity allows for longer contact time, so that you end up getting better response. And so again, this binding affinity will then cause there to be signal transduction or cell signaling, and this leads to cytokine signaling. So cytokine signaling would be the downstream event that's going to happen. We can also get cytoskeleton rearrangement, and this can help with the cell having a change in function. So if you think about like a cell that's have to undergo phagocytosis, receptors on its surface need to be activated, and then it's going to have to rearrange its cytoskeleton to create those pseudopodium that are going to cause the pathogen to be engulfed. So next we're going to look at the B-cell receptor, or the BCR. And so, as I've mentioned, the B-cell receptor has the same structure as an antibody. So there's these immunoglobulin domains. And so if you look on our panels, in panel A, we see this membrane bound form, which is the BCR. Then on panel B, we have our secreted form, and so this would be our antibody. There is also a cytosolic form as it's being produced. And so when the immunoglobulin lacks the carboxyl terminal that has the transmembrane segment, it gets secreted. So the cleavage of that causes that to be secreted. So in the plasma cells, they don't have that transmembrane segment, so they get automatically secreted. So looking a little closer at antibodies, and you may have looked at antibodies in more detail in a different course, but they're going to have quaternary protein folding. So we actually end up having these two heavy chains that come together and then two light chains that come together. And we'll look at more closely what this looks like in our next slide. But the antigen is going to have a specificity to a specific antigen, and that is in a region of both the light and heavy chain, and this is known as the variable region. Okay, antibodies have a number of effector functions or activities, so they can help initiate phagocytosis and complement. So we'll talk about complement later on in the semester when we talk about innate response, but this process can be initiated by antibody binding. Okay. So looking a little closer at antibodies and their structure here in this figure, it's a little bit easier to see in panel A. You can see how we have these two heavy chains and then two light chains. The light chains are smaller, so they are lighter. So if we were to test this, if we were to denature these, they use specific gravity. The light chains aren't as heavy as the heavy chain, so the heavy chain is much longer. So they both have a variable region, which you can see as marked as a V. So there's a VH, that's the variable region of the heavy chain, and VL being marked with the variable region of the light chain. And then we have constant regions. We have a single constant region for the light chain, but then 3 constant regions for our heavy chain. Okay. Another important feature of the antibody is these disulfur bonds. So if you remember back to cell bio or when a class you're talking about protein folding, when proteins fold, disulfur bonds are really important in order to maintain those foldings. So we have disulfur bonds within our different regions, also between our heavy chains, and also between our light chains and our heavy chains. So you can see how important disulfur bonds are. And so you can imagine if there's a mutation to the amino acid here, that could dramatically affect the ability for an antibody to fold. Okay. So it's important to keep in mind that variable region at the end or the top of our antibody is where the antigen is going to bind. And so this is the area that changes and lends to being specific to a specific antigen. Okay. The other thing to keep in mind, and we'll talk about later on in the semester, is there's different forms of light chains and different forms of heavy chains. So we can get different combinations, and different B cells, different types of antibodies would have different combinations of these. To kind of expand upon that, when we look at the different classes of antibodies, we can get different isotypes. So there's immunoglobulin A or IgA, IgD, IgE, IgG, and IgM. And you'll notice that each of these have a different heavy chain to them that's unique. So one of the ways, if we had purified antibodies and we didn't know what class of antibodies they were, what isotype, we could then look at the heavy chain, and depending upon what heavy chain is present, would let us know. So I had alluded to in the last chapter class switching. And so class switching refers to switching at the heavy chain. So a B cell can switch the heavy chain that they're producing, and then so they can go from producing IgMs to IgGs. And this class switching happens because some antibodies are better at binding. So some heavy chains are better at binding than other ones. So IgGs tend to have a higher affinity than IgMs. So if you're like a memory B cell, you like to be producing IgGs versus IgMs. Okay, and then again, also with our light chain, we have isotypes. And again, we will talk about these later on in the semester, so don't, if this went over your head and you're like, just know that there's different classes of antibodies, and the heavy chain is unique to the different classes. So just to give you a visual of the different classes of antibodies, some of them have similar shapes, like the Y shape, the classic Y antibody shape. So IgG, IgD, and IgE are all monomers, where IgA actually dimerizes. So there's kind of like two of these antibodies that come together to form an IgA. And IgMs actually form a pentamer, so 5 of these Y structures come together to form IgM antibodies. Okay, and so with the B cell receptor, just like with the T cell receptor, there's going to be accessory molecules that aid in signal transduction. So here we have our B cell receptor, and then you'll notice that we have these other molecules off to the left of it. And so it's really this complex together that's required in order for signal transduction to happen. So right next to it, you'll notice that we have our Ig alpha and Ig beta. This is referred to, this is an example of a CD, so those cluster of differentiation. So CD79, so, sometimes we have multiple terms for the same thing. One of the things you'll notice is that it has an ITAM. So ITAMs are the immunoreceptor tyrosine-based activation motif. So obviously ITAM is much shorter. So these are really critical in order for the transduction of the signal to occur. Okay. We also have CD19, CD81, and CD21. And so again, those are going to be important in stabilizing the interactions between the receptor and the antigen, and then allowing for a signal transduction to occur so that we end up relaying the signal inside the cell and then cascading of signaling so that we have a response. So again, big picture pieces, it's not just the B cell receptor. There are other pieces and components in order for a signal to occur to get a response. And then with the T cell receptor, it's going to have similar structures to the immunoglobulin domains. So again, it's going to be a dimer that's going to come together, and within those dimers, we have a variable region, a variable domain, and a constant region, constant domain. Okay, and so that the constant region contains the transmembrane region. For the variable region, there's 3 CDRs that will end up forming the peptide-specific binding site. So in that, where those CDRs are, if we change those amino acids, it would affect which peptide, which antigen it would be able to bind. There are 2 types of T cell receptors. There's where we have the alpha and the beta chain coming together. And then we also have the gamma and delta that come together. And each one of these are going to have different antigens that they would end up being able to bind. So the T cell receptor recognizes and binds both the antigen-derived peptide and also the MHC. So there's going to be binding sites that's antigen specific, and there's also going to be binding sites that allow and stabilize the interactions between the T cell receptor and the MHC on the antigen presenting cell or the other immune cell. Okay. The source of peptides can either be endogenous or exogenous processed antigens. And we'll talk more about the T cells, the receptors, and peptide protein sources later on in the semester. So again, there's a number of different accessory molecules that are important for T cells signaling transduction, and we've already alluded to this, but also it's similar to like with the B cells. And so some of these CDs, these accessory molecules, are important for adhesion. You can think of this binding of the receptor, stabilizing the receptor, and also the MHC or other accessory molecules. Some of them are related to signal transduction, and some of them are function as a member of the Ig superfamily. So looking at how the complex of the T cell receptor looks like, we have our figure here. So again, just like with the B cell receptor, we have a lot of other pieces around the cell receptor. So you can see here where we have our T cell receptor, and then we have our peptide, our MHC class 2. We've already talked about CD4 coming in and stabilizing this. If this was a different type of T cell, it would actually be CD8 stabilizing this interaction. And so again, our function for CD4 and CD8 is to increase affinity for the peptide that's going to be binding to the T cell receptor. We also have CD3. So CD3 contains ITAMs. As we saw, ITAMs are important for signal transduction. And then we also have CD28, and CD28 is going to be interacting with either CD80 or CD86. CD86 is on the APC side. Okay, so this interaction between CD28 and CD80 or 86 is important for complete signaling. And we'll talk about later on when we talk about T cells how we need to have a first signal, a second signal, and a third signal in order to get a response. So next we're going to look at PAMPs. I've mentioned PAMPs when we were talking about chapter 1, but keep in mind that the term PAMPs and MAMPs are more or less interchangeable, and for our case as an immunology class, we would, if you refer to PAMPs, I will take that as pathogen-associated molecular patterns, not plant-associated molecular patterns. So as I mentioned before, botanists have a term called PAMPs too, and so some of the newer literature out would use MAMPs just to avoid confusion, and MAMPs would stand for microbe-associated molecular patterns. And so because we as humans are not pathogens, are not microbes, our cells don't have these molecular patterns, and so our innate cells, our innate immune cells are able to pick up on these molecular patterns and distinguish them from us. So this leads to that tolerance that we had talked about, self versus non-self. Okay, and so our immune cells would have receptors for PAMPs or MAMPs, and so there are MAMPs that are shared between microbes, between bacteria, and then there are ones that are unique to certain types of bacteria. Okay. So some of these receptors are integrated into the membrane, so they would have surface interaction with extracellular MAMPs. Other ones are intercellular, so the microbe would actually have to be ingested and then would interact with the receptor inside the cell. Okay, but again, the MAMPs represent motifs that reoccur, these reoccurring patterns on bacteria, yeast, and parasites. One of the journal club papers we'll probably look at looks at different molecular patterns and which receptors are being activated by them. And so if we look at this table from the textbook, it looks at some of the different receptor families. One of the most common ones that are research looked at are toll-like receptors, TLRs, and there are a number of different TLRs, toll-like receptors. So again, there's a journal club paper that I've done before that looks at these different toll-like receptors and their activation. This is also a concept we'll talk about with the innate response later on in the semester. So different toll-like receptors can be found on the plasma membrane, where other ones would be found inside endosomes or lysosomes, because after a cell has undergone phagocytosis, some of the material would be found, some of the pathogen material would be found in these structures. Okay, the ligands that will trigger toll-like receptors could be carbohydrates, lipoproteins, flagellin proteins, and so it all depends upon what toll-like receptor you're looking at, where it's found, and what the ligand is. But the end result usually ends up being that you have production of antimicrobials or antivirals, and then you can have cytokine production that leads to activation of inflammation. We also have these C-type lectin receptors, so CLRs, and these are found in the plasma membrane and can interact with carbohydrate components from fungi and mycobacteria, viruses, parasites, and some allergens. And again, activation of inflammation, production of cytokines are pretty standard. There's additional pattern recognition receptor families, as you can see on this chart, but again, TLRs and CLRs are the ones that are studied more widely, not to say that the other ones aren't. So as we just mentioned, with these PAMP receptors being signaled and activated, or by having our B cell or T cell receptor activated, we can get cytokine production and release, and those cytokines then can bind to their receptors. Okay, so just like we saw, ligand receptor binding with our B and T cell receptor, the cytokines are going to be the same. The cytokines will act as the ligand and bind to a receptor that's bound to the cell, to another immune cell or the same immune cell in which the cytokine was produced. We'll look at a little bit about that in just a moment. So then we get the cytokine receptor binding. This is non-covalently, but there can still be a high affinity that occurs. Okay, from that cytokine signaling, we're going to end up getting induction of changes in transcription programming of the target cell, and this can lead to changes in its metabolic and proliferative state, so that we can get immune cells that will actually proliferate and then provide their function. So if we have a T cell, and T cell will have an IL-2 receptor, and if IL-2 binds that receptor, that causes signal transduction and causes the T cell to actually proliferate and undergo clonal selection, just as an example. So there's some general properties of cytokines and chemokines. So they can act in several different ways. So there's endocrine action, paracrine action, autocrine action. So endocrine action is where it's going to be released into the bloodstream, and then it's going to affect distant cells. So cytokines and chemokines, even if they're being produced because you cut your finger, they can actually recruit in cells through the bloodstream and tell them, like, oh, this is where you need to go. Okay, where paracrine action is going to be released to nearby cells. So if we have a T helper cell trying to help a B cell to differentiate into a plasma cell, it can produce cytokines that will have that local nearby effect to the B cells directly near it. And then we can have autocrine action, and this is where a cell would actually release a cytokine, and it's going to bind it on its own receptors and then cause signal transduction. So my example with IL-2 with T cells, so IL-2 can be produced by the T cell, and then the T cell will actually bind that, and it kind of causes a feedback loop to cause the cell to become activated. So the way in which cytokines can affect neighboring cells, can be termed in several different ways. I highly do recommend looking at the next slide, or the figure 3-18, as I go through these definitions. So this is kind of the text slide to accompany the next slide. Pleiotropic, this is where the cytokine or chemokine can have activity that induces different biological effects depending upon the target cell. So it can actually work on a number of different cells, and each cell could have a different function. Redundant is where we could have multiple cytokines that are going to act on the same target cell and have similar effect. Synergic is where we're going to have a combination of the two cytokines that cause a greater additive effect. And anergistic is where we're going to actually have one cytokine inhibiting the other cytokine's effect. So it's a way to override one cytokine from the other. And then we can get this cascade effect where cytokines are going to target one cell, but then they produce an additional cytokine that causes a cascading effect. So we'll look at each one of these in our figure. So here we can see all these different terminologies in this figure. On panel A, we have our effect. So we're focusing on our T helper cell. Just keep in mind that the T helper cells are not the only ones that are producing cytokines and having effects of cytokines. So we could substitute this for a lot of other examples. It's just that the author of the textbook has selected T helper cells. So for pleiotropic, this is where our T helper cell is going to produce IL-4. IL-4 or interleukin-4 can affect a number of different immune cells, such as B cells, cytotoxic T cells, and mast cells. And you can see that each one is going to have a slightly different effect. So yes, we have proliferation occurring, but for our cytotoxic T cell precursor, it's going to differentiate into a mature effector cell. And for our B cell, we get activation, proliferation, and differentiation occurring. For redundancy, again, we have our T helper cell here, and it's going to produce a number of cytokines, and all of those cytokines are going to cause proliferation of the B cell. So we have IL-2, IL-4, and IL-5, but you can kind of think of this, we really want to make sure this happens, and so maybe this B cell is somehow deficient in IL-4 receptors, and so we want to make sure that it's still getting that signal. You need to proliferate, okay? Synergic, this is where we're going to have two cytokines, so like cytokine IL-4 and IL-5, and both of these added together, when we have both of them present, actually causes the B cell to induce class switching. So where if we just had one of them present, we would just get proliferation, but because we have the two together, it tells the cell to do something different. Okay, it's like, if you had flour, maybe you'd make a cake, but if you have flour, sugar, and chocolate chips, you'd make cookies, right? So the recipe, the product changes, the effect changes. For antagonism, this is where we're going to have two cytokines and they actually work against each other. So here we have IL-4, which normally would cause proliferation, but if we have interferon gamma being produced, it actually blocks the proliferation, blocks any kind of class switching from occurring in the B cell. Okay, and then the last kind of effect we can have, or cytokine effect that we can have, is this cascade induction. So we can see here we have this activated T cell. It's going to produce interferon gamma. That's going to act on the macrophage. The macrophage then produces IL-12. That then affects the activated T cell, so it's going to produce more interferon gamma, TNF alpha, IL-2, and other cytokines. So again, this kind of cascading, one event has to happen in order for the next event to happen, which then causes another. And again, this is just a way to have feedback signaling, so we don't, so the immune system doesn't go the wrong path. So there's 6 major cytokine families, and we'll look at some of these different cytokine families in more detail. I'm not going to like say, which, here's a cytokine, what family is it a member of, though some of it's kind of intuitive. So if you see something with IL-1, then we could assume that it's in the interleukin-1 family or IL-1 family. Okay, so this table is just included just to kind of give you an overview of the different families. And again, we're going to look at some of these families more closely, but this is just meant to kind of introduce you to cytokines and what their role is, and this inflammation is the word. So keep that in mind. Okay, and so looking at interleukin-1 family, these cytokines are going to be prone to promoting inflammation. So they get IL-1 gets stimulated when we have viral, parasitic, or bacterial agents present. So they're going to get secreted out, and they get secreted early, so during the innate response by macrophages and dendritic cells. They can act locally by causing capillaries to become more permeable. So this actually allows for other leukocytes to come into the area where we have an infection, again, whether it's a viral, parasitic, or bacterial infection. And so this can also help activate adaptive immune response also. So our next family is the class 1 cytokine family. So these are very diverse in their action and their cell target. And so one of the things to keep in mind is that with this family, they're usually made up of multiple subunits. So you can see on the table here some of the common subunits that are in class 1 family receptors. And so you can see some of the examples. Throughout the semester we'll talk about IL-2, and IL-6 will be a big one that we'll talk about, because IL-6 has been shown to be a critical cytokine for severe cases of COVID. So we'll talk a little bit about IL-6 through the journal clubs and through the course of the semester. So class 1s are important, but they're a little bit, because they're so diverse, they're a little bit tougher to get your head around. Then for our class 2 cytokine family, we have the interferons. So the interferons fall into 3 different categories or types. So type 1 interferon would be interferon alpha and beta. So both of these are dimers, and they have antiviral effects. They get secreted by activated macrophages and dendritic cells. So again, they're very important in the innate response, and they're going to induce synthesis of ribonucleases that will break down RNA and also inhibit they also inhibit protein synthesis. So again, if we have a viral infection, it's really important that's 2 ways in which we can combat a viral infection, because a virus is going to cause our cells to produce proteins for the virus, and also there will be additional RNA around, because even if it's a DNA virus, it has to go through the RNA, produce RNA before it can make protein. And so by kind of shutting this down in the cell by the production of interferon alpha and beta, they would have antiviral properties. For type 2 interferons, this would be interferon gamma. Again, we have a dimer here, and they're going to be produced by activated T or natural killer cells. And so interferon gamma is really important in modulating how the adaptive immune response occurs. So whether we're activating or deactivating cells. Type 3 interferon family is interferon lambda. And so this is secreted by dendritic cells, and this upregulates genes that control viral replication and host cell proliferation. So I would say for the interferons, you should know there's 3 types, and you should know which interferons are found in each of those types. And then we have TNF family, tumor necrosis factor. So it's going to regulate the development and effector function along with the homeostasis of cells in both the skeletal, neural, and immune system. So this is a factor that's not just immune system. Okay, so it has multiple functions. TNF cytokines are both soluble or membrane bound. So important thing to keep in mind. So here we can see how we have in our figure here, we have our receptor. When it binds TNF, that actually causes this trimerizing happening, and that actually activates NF-kappa B. So if you've talked about cell signaling, you might have talked about NF-kappa B. NF-kappa B is a really important cell signaling molecule in the immune system. And so it actually leads to cell survival and other downstream events. So with the TNF family, there's several different variants of it. So TNF alpha is probably the most popular one or commonly studied one. So it has pro-inflammatory responses, and it gets produced by activated macrophages. So one way to check whether a macrophage has been activated is to see how much TNF alpha is being secreted. Okay. We also have TNF beta. That's going to be produced by activated lymphocytes, and this helps deliver signals to other leukocytes and endothelial cells. Lymphotoxin B is another TNF member, and so it helps with lymphocyte differentiation. Some other TNF members that we'll talk about in the course of this semester is BARF and APRIL. So those are important when looking at B cell development and homeostasis. So again, we'll talk about those later on in the semester. CD40 ligand or CD40L is important for T cell differentiation, signaling to B cells. So again, we'll talk about those later on. And then Fast ligand, you might have heard about in another course because it's important to induce cell death through apoptosis. And so later on in the semester, when we talk about how these cells get, how bad lymphocytes get told to die, we'll talk about fast ligand binding. So our next family that we're going to look at is the interleukin 17 or IL-17 family. So again, these are pro-inflammatory molecules, and they can be expressed by a variety of number of cells. So there are receptors for interleukin 17 found on neutrophils, keratinocytes, and other non-lymphoid cells, so other non-immune cells. So they actually tend to work to have communication between innate and adaptive immunity, and generally they're going to be in homodimers, so single ones, and as transmembrane proteins. So this table just shows some of the different family members of the interleukin cytokine family. Again, I don't expect you to memorize this table. What I'd expect you to take home is that if you look at the different cell types that express these cytokines, you can see that there's quite the range. And then if we look at the effect, generally this is going to be pro-inflammatory. So for chemokines, these are going to help to direct where leukocytes are going to migrate to. So again, imagine you have an injury and you need to recruit in immune cells to help, and whether this is like an injury, you cut your finger, or whether this is an infection going on in your pancreas, we're going to want to make sure we can recruit in additional immune cells into that location, because even if we have resident macrophages in that location, we're going to want to have other immune cells, other leukocytes. Okay, and so chemokines become this kind of chemical signal in which immune cells can use to traffic to a location. And so obviously wherever the site is of injury or infection is going to have a higher concentration, and so this causes the cells to actually migrate towards the highest concentration of them, and that's kind of how they end up knowing where to go, because they're going to have receptors for these chemokines. So it's almost like they can, you can think of it like they smell them. So it's kind of like the cookie trail, right? So, if someone's baking cookies, you can smell the cookie, or in my neighborhood, everybody's like barbecuing, and so like someone's got the smoker on, and oh, where's that barbecue? And you're gonna go and you're gonna follow, and when the smell gets stronger, you're in the right location. And so chemokines allow for the immune cells to do the same function. I'm not going to ask you about the residues and their locations, but just be aware that some of the naming of these chemokines are based off of their sequence and their structure. So in general, the chemokine receptors are going to be G protein coupled receptors. So if you've taken another course and you talked about cell signaling, you probably talked about G protein coupled receptors. So these are transmembrane receptors that have 7 passes through the membrane. And they're going to be coupled with G proteins, GTP and GDP. So they're going to bind those, and signaling happens based off the phosphorylation. That phosphorylation then leads to further downstream activation and can cause changes in gene expression. So for these chemokines, so some of them are able to bind more than one receptor. And then other cases, the receptors can actually bind more than one chemokine. So it allows for some redundancy so that, you get the leukocyte migrating to that location. So when we look at receptors in cell signaling, just in case it's been a while since you took a course that talked about cell signaling, we want to keep in mind that we're going to have a receptor, and that's going to bind to the ligand. So in order for the signal to be generated, we have to get binding of a ligand to its receptor. And generally this is going to be a cell-bound receptor. It could be either be a cell surface receptor or inside the cell in a membrane. Okay, and so a cell can change how susceptible it is based off of the expression of receptors. So if you have a small amount of receptors, even if there's a large amount of ligand, you can only bind so much, right? So you're not going to get as strong of a response unless this is like an all or nothing, right? Where if we increase the amount of receptors present, so we increase the expression, we would be able to bind more ligand and get a stronger signaling event. So in most cases, cell signaling is going to induce changes in transcription, and through the changes in transcription, we're going to get changes in protein, and then in many cases, it could be proliferation or differentiation of the cell. So, we could have gene expression where we now produce because one cytokine has bound to the receptor, we're going to actually get production of another receptor, so that we can actually sense that ligand being present. Sometimes multiple signals go through multiple receptors in order to get a certain outcome. So you need, receptor A, B, and C to bind before you actually get the outcome. Because again, if just one thing is right, one thing is being activated, that's not sufficient to actually warrant that change. Okay, so it's really this integration of all the signals that are coming from the cell, because we can also have signals that are actually inhibiting the effect from happening, right? So we can have these antagonists. So we're integrating all this information to do we end up having an effect inside the cell? And, you can't talk about cell signaling and not show some scary diagram where, again, we have this ligand, we have receptors, we have associated molecules to the receptor so that we're getting phosphorylation. That phosphorylation causes activation of kinases. So downstream, we end up getting activation of genes and protein synthesis. Okay, so there's many different pathways that can lead to this, and there's many different accessory proteins involved in this. Okay, so this is actually showing tyrosine kinase receptor. We were just talking about a G protein coupled receptor, so there's different types of receptors, so lots of different options. So even though I've already kind of foreshadowed about some of the receptor and cell signaling that happens in the immune system, I just want to go through some examples. So we had talked about the B cell receptor and how it will actually dimerize and then even multimerize together, so they'll get this receptor clustering, and that receptor clustering, when antigens bound, leads to that signal transduction occurring. A lot of times this clustering of receptors occurs on lipid rafts. We can also get tyrosine phosphorylation. So this is an early step that's found in many signaling pathways. So for T cells, CD3 acts as a tyrosine site for tyrosine phosphorylation. And then in the B cells, it's that IgA and Ig beta. So that's CD79 that gets phosphorylated on their ITAMs, right? And as I mentioned before, ITAM stands for immunoreceptor tyrosine activated motif, and so this is the site where we can get phosphorylation. So really these are acting as adapter molecules. So we can also have Sark family kinases that phosphorylate the tyrosine. So these kinases are going to become activated themselves when phosphorylated. And then in these cell signaling pathways, we also have adapter proteins. So the adapter proteins help to gather the different members of the cell signaling pathway. So, we have PIP2 and PLC gamma. And so our adapter protein is going to kind of hold these in locations so that they're nearby each other. We can also have RAS or MAP kinase cascading activation. So here we have RAS acting as the G protein, and so when it becomes activated, it causes further downstream activation of MAP kinase. And so here we see this MAP kinase in this box where we have this MAP kinase activation. So this would lead to phosphorylation of ERK. We can also have Jun protein, phosphorylation of Jun and Phos. And so then downstream, we would get IL-2 activation. So activation of IL-2 gene, so we get production of that. So another common cell signaling pathway is PKC or phosphokinase C, and that activates NF-kappa B, nuclear factor kappa B. So NF-kappa B is normally inactivated in the cytoplasm because it binds an inhibitor. But then when phosphokinase C becomes phosphorylated, it actually causes a dissociation of the inhibitor NF-kappa B. So NF-kappa B can become activated and translocate into the nucleus, where it will actually activate transcription. The reason why I bring all of this up and all these cell signaling pathways is that in our journal club papers, we will most likely at some point see some of these different phosphorylations and some of these different signaling pathways. So we might have a paper where they're going to look at phosphokinase C, or they might look at NF-kappa B. And so again, ERK, phosphorylation of ERK. So I just again want to introduce the idea that sometimes when these players when these molecules come into play, it's because they're looking to see whether when we have a ligand, when we have a receptor, whether we actually get activation occurring. Okay, so in no way am I saying memorize these cell signaling pathways. What I'm saying is just appreciate them for now. They will come back when we talk about activation of these different cells, and again in the journal club papers. So as mentioned, there can be many different effects that can happen when we have antigen signaling, right? So when antigen binds to the receptor, some of the different effects we might have is that it will actually cause dendritic cells to move to the required location where they're needed. Macrophages and neutrophils can upregulate the phagolysosome activity. This is an important structure when we talk about phagocytosis. So it allows for macrophages and neutrophils to undergo phagocytosis better. It can cause them to cause cytokine production. And then with dendritic cells, it can cause them to upregulate MHC class 1 or class 2 so that they can present antigen peptides. For cytoplasmic proteasomes, this can cause more effective processing of antigens into peptides. So looking at this chapter, we have the recognition part. The recognition is where we're having ligand binding to receptors. And so, we have the antigens binding to receptors like your T cell or your B cell receptor, but we also have cytokines binding to their receptors also. The other part to this chapter is looking at response, and so that response really goes to signal transduction. And we do want to keep in mind that signal transduction is a very complex subject, and lots of memorization, lots of different pathways depending upon what receptor is being used. And so again, you can take a whole semester, a whole course on cell signaling. And so really, let's look for similarities and differences. Let's what pieces can we generalize, and then build upon that. So both B and T cell lymphocytes have similar strategies once the process is underway, and we'll talk about that later on in the semester when we talk about adaptive immunity and how B cells and T cells mature and become activated. But the difference is rely on the level of the receptors used, right, in early membrane-linked events. So the nuances between that. But definitely by understanding antigen receptors and these earlier events, the differences, we can better understand how signaling activation works for each of these different cell types. And again, this is information that's going to be expanded upon throughout the whole entire semester. So if you feel overwhelmed, don't worry about it. It's just about laying a foundation and then moving on and building more detail into that foundation.